Thank you, everybody. For our first of several exciting talks, we have Dr. Juan Biancotti from Johns Hopkins University in Baltimore, Maryland. Is Dr. Biancotti? Oh, he's coming. It's a big room. And he will be presenting on transcriptome analysis reveals proximal airway epithelial and pulmonary mesenchymal dysregulation in human and murine fetuses with diaphragmatic hernia. Dr. Biancotti.
Good morning, and thank you for the opportunity to present our results. As you all know, congenital diaphragmatic hernia leads to lung hypoplasia, pulmonary hypertension. The underlying molecular pathway responsible for lung hypoplasia remain poorly understood. Correlation of mouse model with the human disease has not been comprehensively determined. Nitrofen BPCA, which is the drugs that we use to generate the mouse model, generate both type of embryos in the same mouse, embryos with defect and without defect. The purpose of our work is to analyze gene expression profile within human and murine fetal CDH lungs at the pseudoglandular canalicular stage of development. We collected E15.5 mouse lung embryos exposed to nitrofen BPCA and control ones. Also collected the 21-week human lung fetuses to control and to CDH from an NIH neurobiobank. Isolated total RNA, make a library preparation, and send for sequencing. The gene set enrichment analysis was performed by differential expression. We also use enrich and David web servers to do further analysis. CDH mouse embryos had a significantly lower lung over body weight ratio compared to control and even to embryos that didn't have the defect. On the left, we see the distribution of control and CDH mouse samples by principal component analysis. On the right, we see a volcano plot that shows us that there is a much higher number of upregulated than downregulated genes in CDH. To validate the results of RNA-seq, we selected a set of genes, upregulated genes, that were involved in the lung development and measured the gene expression by RT-qPCR. As you can see, all of them were upregulated as well. The left side graph, each bar represents a term, and the length of the bar, the p-value. The top left, it shows that proximal airway ciliated and club cells are the most significantly enriched cell types. On the right, the functional cluster annotation shows us that cilium, muscle proteins, and extracellular metrics are among the most significantly affected clusters. Bottom left, we identified two transcriptional factors, FOXA1 and CBP-A, that are involved in lung development, and others that were involved in myogenesis. For human samples, we can see the distribution again in the principal component analysis on the left, and on the right, the volcano plot showing that also for human samples, we have a bigger number of upregulated than downregulated genes in CDH. We also validated RNA sequencing results by RT-qPCR. And on the top left, there is a graph for gene ontology for biological processes that shows enrichment for genes related to cilium and collagen-containing ECM. In the graph below, the human phenotype ontology, we see that there is a strong enrichment in alteration of respiratory epithelium, which correlates with the specific cell types, where we can see on the bottom right, proximal airway epithelium is very highly enriched. And regarding transcription factors, we identified 2 transcription factors, ELF3 and SP2, as being significantly upregulated, that are involved in proliferation and differentiation of epithelial cells during lung development. So in conclusion, from the transcriptome perspective, the nitrophen BBCA mouse model with defect of CDH mimics CDH disease in human fetuses. The pathogenesis of pulmonary hypoplasia and pulmonary hypertension in fetal CDH may involve the upregulation of genes associated with proximal airway epithelial and lung mesenchymal development. This data may contribute to the design, we hope, of novel molecular targets to help reverse aberrant development within the fetal CDH lung. Thank you for your attention. I'll be happy to answer any questions.
Open this up for questions.
Zani Sakits, Toronto. Really enjoyed your paper. The model is not an easy model, and as we know, not necessarily you have homogeneity in the population of pups that develop CDH. So my question to you is whether you were able to phenotype those pups, those lungs, before you actually sequenced.
Yes, when we isolated the lungs, we selected only those, or we grouped them between those with defect and separated those that didn't have the defect.
But do you know if the degree, the severity of pulmonary hypoplasia that you had was the worst spectrum, or?
It was because the presence of the hole in the diaphragm.
Okay.
Those that have the defect, but we don't know exactly if it is a higher phenotype, a worse phenotype because of the teratogens, or because there is an additional effect of the pressure induced by the liver and organs going inside the cavity, thorax cavity.
Thank you. Great work.
Sure. Thank you.
I have a question. So when you were looking at the differential gene expression, and it's different in the mouse and the human on the level of the surfactant, as I understand, how do you think that makes it, does that make a lower fidelity model, in your opinion, for the mouse, or higher?
Sorry, could you repeat the question? I couldn't hear very well.
With the difference in surfactant expression, as I understand, do you think that that makes the mouse model more or less valid for you?
We consider that the mouse model parallels quite well, at least from the gene expression perspective, the human disease. So phenotypically, it also shows defect. So I think it's a good model to use for the study.
Thank you so much.
Thank you.